Assalamu alaikum friends. Friends, hope so you all will be fine. Friends, today's topic of our discussion is glycolysis. These are the outlines of glycolysis. In this video, I will cover important aspects, steps, regulation, and energetics of glycolysis. In next video, I will explain last two steps of the glycolysis. So let's start our lecture with the important aspects of glycolysis. Okay, important aspects of glycolysis. Students, the glycolysis process it occurs in all cells of human body. Second one is this process proceeds aerobically and anaerobically. Third one is it is important source of energy for brain and matured RBCs. The glycolysis process occurs in all cells of human body, and this process proceeds aerobically and anaerobically. Aerobically means in the presence of oxygen and anaerobically means in the absence of oxygen. And it is an important source of energy for brain and matured RBC. Matured RBCs have no any mitochondria, so for energy production it depends upon glycolytic pathway. So coming toward the fourth point, during sternous exercise, the demand of oxygen increases in our body. During sternous exercise, the demand of oxygen increases in our body, but there is a low supply of oxygen toward the skeletal muscles. And at that condition, the glycolytic pathway proceeds anaerobically. Students, during strenuous exercise, demand of oxygen increases in our body, but there is a low supply of oxygen towards skeletal muscles, and at that condition, the glycolytic pathway proceeds anaerobically. Now, coming toward the fifth point, the glycolytic activity it is very low. The glycolytic activity it is very low in cardiac muscle. Okay, students. And coming towards sixth point, that is, it is an important pathway for the production of carbon skeleton of non-essential amino acid. And last one is this process involves in gluconeogenesis. Gluconeogenesis means production of glucose from non-carbohydrates precursors. स्टूडेंट्स मैं इनको रिपीट कर देता हूँ. अगर ग्लाइकोलाइसिस को डिफाइन किया जाए, ग्लाइकोलाइसिस की डेफिनेशन है ऑक्सीडेशन ऑफ ग्लूकोज टू पायरुवेट and lactate formation. This is the definition of glycolysis, that oxidation of glucose to pyruvate and lactate formation. This is called as glycolysis. The important aspects of glycolysis are: this process occurs in all cells of human body, and glycolysis process proceeds aerobically and anaerobically. It is an important source of energy for brain and matured RBCs. During stillness exercise, during hard exercise, the demand of oxygen increases in our body, but there is a low supply of oxygen towards skeletal muscles, and at that condition, the glycolytic pathway proceeds anaerobically. The glycolytic activity is very low in cardiac muscles, and this glycolysis process, it is responsible for the production of carbon skeleton of non-essential amino acid, and this process also involves in the gluconeogenesis, means the synthesis of glucose from non-carbohydrate precursors. Students, these all are the important aspects of glycolysis. Now coming toward the steps of glycolysis. Steps of glycolysis. Students, glucose by action of hexokinase or glucokinase enzyme, and ATP is utilized here. ATP converts into ADP and inorganic phosphate. This glucose, it converts into glucose 6 phosphate, and phosphate group gets attached to the sixth carbon of glucose and converts into glucose 6 phosphate. In next reaction, by action of isomerase enzyme, this glucose 6 phosphate, it converts into fructose 6 phosphate. Okay. In next reaction, by action of phosphofructokinase, phosphofructokinase, and here ATP is utilized, and ATP converts into ADP and inorganic phosphate, and fructose 6 phosphate. Now phosphate group gets attached to the first carbon of fructose. It converts into fructose 1,6 bisphosphate. Okay, students, these steps, first, second, third, these steps, they are the part of preparatory phase of glycolysis. So here is the this is the this is the preparatory phase of glycolysis. Okay. Now fructose 1,6 bisphosphate, with the help of aldolase enzyme, it converts into first dihydroxy acetone phosphate, which then converts into glyceraldehyde 3 phosphate with the help of isomerase enzyme. ओके स्टूडेंट्स, एडुलस एक हैमर, हथौड़े की तरह काम करता है और इस कंपाउंड को दो टुकड़ों में कन्वर्ट कर देता है. इट इज अ 3 कार्बन कंटेनिंग कंपाउंड, इट इज अ 3 कार्बन कंटेनिंग कंपाउंड. अब इस ग्लाइसरॉल्ड 3 फास्फेट के दो मॉलिक्यूल्स हम लेकर के आगे अपने पाथवे को कंटिन्यू करेंगे. First reaction of glucose by action of hexokinase or glucokinase enzyme, it converts into glucose 6 phosphate. In next reaction by action of isomerase enzyme, it converts to fructose 6 phosphate. With the help of phosphofructokinase enzyme, it converts into fructose 1,6 bisphosphate. With the help of hydrolase enzyme, it converts into first dihydroxyacetone phosphate, which converts into glyceraldehyde triphosphate with the help of isomerase enzyme. अब इस glyceraldehyde triphosphate को टू मॉलिक्यूल्स कंसीडर करके हम अपने नेक्स्ट पाथवे को प्रोसीड करेंगे. नेक्स्ट रिएक्शंस, glyceraldehyde triphosphate, glyceraldehyde 3-phosphate with the help of glyceraldehyde 3-phosphate dehydrogenase enzyme. Here NAD plus, it converts into NADH and H plus. It converts into 1,3 bisphosphoglycerate. In next reaction, by action of bisphosphoglycerate kinase, it converts into 3-phosphoglycerate. And here there is a production of ATP. क्योंकि हम टू मॉलिक्यूल्स कंसीडर करके चल रहे हैं तो यहाँ पर भी टू मॉलिक्यूल्स आएंगे और यहाँ पर भी NADH and प्लस के टू मॉलिक्यूल्स. Okay students, now 3-phosphoglycerate with the help of mutase enzyme, it converts into 2-phosphoglycerate. And next reaction, by removal of water molecule with the help of enolase enzyme, it converts into phosphoenol pyruvate. And in last reaction, by action of pyruvate kinase, it converts into, and there is production of ATP, ADP, there is production of ATP and pyruvate. So students, these are the steps of glycolysis, and this is the oxidative phase of glycolysis. This is the oxidative phase of glycolysis. This glyceraldehyde 3-phosphate के two molecules consider करके चल रहे हैं. Glyceraldehyde 3-phosphate with the help of dehydrogenase enzyme NAD plus converts into NADH and H plus. It is an electron carrier molecule. In electron transport chain, one molecule of NADH and H plus, it gives 2.5 ATP, while two molecules they give 5 ATP. Once bisphosphoglycerate with the help of bisphosphoglycerate kinase enzyme, there is a production of ATP. It converts into 3-phosphoglycerate. With the help of mutase enzyme, it converts into 2-phosphoglycerate. And with the help of enolase enzyme, there is removal of water molecule. It converts into phosphoenol pyruvate. And with the help of pyruvate kinase, there is production of ATP. It converts into pyruvate. And this is the oxidative phase of glycolysis. This is the preparator phase, and this is the oxidative phase of glycolysis. Students, some important points in glycolysis you must need to remember that first step, this third step, and last step, they are irreversible, while other steps they are reversible. They are reversible. Okay. The first step, this is the first step, this is the second step, this is the third step, this is the fourth step, this is the fifth step, sixth step, 7, 8, 9 and 10. So the first step, third step and last step, these are irreversible steps, while other steps they are reversible. Now this step catalyzed by phosphofructokinase, it is the slowest step in glycolysis, and it is a committed step because when fructose 6-phosphate converts into fructose 1,6 bisphosphate, then ultimately it leads to production of pyruvate. It is a 3 carbon atom containing compound, pyruvate. And the steps catalyzed by pyruvate kinase enzyme and bisphosphoglycerate kinase enzyme, they are substrate level phosphorylation step. What does it mean, substrate level phosphorylation? Means production of ATP from substrate without undergoing into electron transport chain. So it's now coming toward the production of energy from glycolytic pathway. First of all, from aerobic pathway of glycolysis, aerobic pathway. Now, in oxidative phase, two molecules of NADH and plus they are producing. In electron transport chain, two molecules of NADH and plus they give 5 ATP. They give 5 ATP, and you can see here there is production of 2 ATP and there is production of 2 ATP here. There is production of 2 ATP and here there is a production of 2 ATP, and there is you can see, and 2 and 2 4 ATP. So total is 9 ATP. Okay. Friends, यहाँ से हमने 1 ATP here, 1 ATP here, there are 2 ATP in preparatory phase. They are utilizing. हमने इन्वेस्टिगेट थे इस पाथवे में. अब जो इन्वेस्टिगेट थे वो दोबारा से रिटर्न भी तो करने हैं ना. So 9 ATP minus 2 ATP, there are total amount of ATP 7 ATP. So the net production of ATP, there is energetics. The total amount of ATP production from aerobic pathway, from this is aerobic pathway, from aerobic glycolysis, the total amount of production of ATP is 7 ATP. मैं अगेन रिपीट करता हूँ. यहाँ से देखिए टू मॉलिक्यूल्स NADH एट प्लस प्रोड्यूस हो रहे थे. इलेक्ट्रॉन ट्रांसपोर्टेशन में जाकर के ये एक मॉलिक्यूल 2.5 ATP देता है और टू मॉलिक्यूल 5 ATP देते हैं. एंड टू मॉलिक्यूल्स 2 ATP यहाँ पर, 2 ATP यहाँ पर, 2 and 2 4 ATP, 4 and 5 टोटल 9 ATP, और 2 ATP यहाँ से हम इन्वेस्ट किए थे, अब रिटर्न करने हैं. सो -2 7 ATP एरोबिक पाथवे ऑफ ग्लाइकोलाइसिस से 7 ATP प्रोड्यूस होते हैं. अब एरोबिक ग्लाइकोलाइसिस में एक ग्लूकोज मॉलिक्यूल पायरुविक एसिड के पायरुवेट 2 मॉलिक्यूल्स में कन्वर्ट होता है, लेकिन एनारोबिक ग्लाइकोलाइसिस में एक ग्लूकोज मॉलिक्यूल पायरुवेट में कन्वर्ट होता है और दूसरा लैक्टेट में. अब होता है क्या, लैक्टेट कैसे बनता है? जो पायरुवेट है ये कन्वर्ट हो जाता है लैक्टेट में विद द हेल्प ऑफ लैक्टेट डिहाइड्रोजिनेज एंजाइम. और यहाँ पर ये जो NADH एंड H+ प्रोड्यूस हो रहे थे ये यहाँ पर यूटिलाइज हो जाते हैं. NADH एंड H+ ये यूटिलाइज होकर NAD+ बना देते हैं. तो इस तरह से लैक्टेट की फॉर्मेशन हो जाती है. अब देखें, क्योंकि NADH+ एंड H+ तो यूटिलाइज हो गए, तो टोटल अमाउंट ऑफ एटीपी कितना हुआ? 2 and 2 4 ATP यहाँ से हो गए 4 ATP, एंड 2 ATP चूंकि इन्वेस्ट किए थे अब रिटर्न करने हैं -2. So total amount of ATP production from anaerobic pathway is 2 ATP. From this is the production of ATP from anaerobic pathway of glycolysis. The total amount of ATP production from anaerobic glycolytic pathway is 2 ATP. क्योंकि पायरुवेट एक मॉलिक्यूल जब लैक्टेट में कन्वर्ट होगा तो NADH and H+ मॉलिक्यूल यहाँ पर यूटिलाइज हो जाएंगे और लैक्टेट की यहाँ पर फॉर्मेशन हो जाएगी. तो total amount of ATP production is 2 ATP from anaerobic pathway. Students, now coming toward the regulation of glycolysis. Regulation. Regulation um, regulation. Students, the hexokinase enzyme, the hexokinase enzyme It is inhibited by glucose 6 phosphate. Glucokinase enzyme, it is activated by insulin and inhibited by glucagon. Okay. Now phosphofructokinase, it is activated by insulin and inhibited by glucagon. And this phosphofructokinase allosterically, एलोस्टेरिकली क्या मतलब देखिए स्टूडेंट्स, एंजाइम के पास एक्टिव साइट भी होती है, एलोस्टेरिक साइट भी होती है। एलो मीन अदर, स्टेरिक मीन साइट, एक एक्टिव साइट और दूसरा एलोस्टेरिक साइट। एक्टिव साइट पर सबस्ट्रेट आकर बाइंड करेगा, एंजाइम सबस्ट्रेट कॉम्प्लेक्स की फॉर्मेशन होगी और प्रोडक्ट की फॉर्मेशन हो जाएगी। एलोस्टेरिक साइट पर एलोस्टेरिक एक्टिवेटर इनहिबिटर आकर बाइंड होंगे। जब एक्टिवेटर आकर बाइंड हो जाएंगे तो एंजाइम एक्टिविटी को बढ़ा देंगे, जब इनहिबिटर आकर बाइंड हो जाएंगे तो इसकी एक्टिविटी को कम कर देंगे। इसको कहते हैं एलोस्टेरिक साइट, एलोस्टेरिक इफेक्टर्स। तो क्या हुआ कि एंजाइम के पास दो तरह की साइट्स हैं। अब डिपेंड करता है, जब एक्टिवेटर आकर बाइंड करेगा तो एंजाइम की एक्टिविटी इनहांस कर देगा, जब इनहिबिटर आकर बाइंड करेगा तो क्या होगा, इसकी एक्टिविटी को रिड्यूस कर देगा। तो फॉस्फोप्रोटोकाइनेज allosterically it is activated by AMP, adenosine monophosphate, and fructose 2,6 bisphosphate. And allosterically it is inhibited by ATP and citrate. Okay, this was one, first one, second one, and third one is pyruvate kinase. Pyruvate kinase, it is activated by insulin and it is inhibited by glucagon. Students, it is regulation of glycolysis. Hexokinase enzyme, it is inhibited by glucose 6 phosphate. Glucokinase enzyme, it is activated by insulin and inhibited by glucagon. Phosphofructokinase, it is activated by insulin and inhibited by glucagon. And allosterically, it is activated by AMP and fructose 2,6 phosphate and inhibited by ATP and citrate. And pyruvate kinase, it is activated by insulin and inhibited by glucagon. So students, it is all about the important aspects of glycolysis, steps of glycolysis, regulation of glycolysis, and energetics. In my next video, I will discuss about the remaining part of glycolysis, and that is hexokinase and glucokinase difference and inhibition of inulase enzyme by fluoride bulb. Keep watching and thank you.